Good afternoon and welcome to another Research in Action brought to you by the Division of Research at Florida Atlantic University. My name is Karen Scapinato. I will be today's moderator. With that, it is my very great pleasure to introduce today's speaker. Dr. Shirley Pomponi is a research professor at our Harbor Branch Oceanographic Institute. She earned her Ph.D. in biological oceanography from the University of Miami Rosenstiel School of Marine and Atmospheric Science, and is an ocean explorer and marine biotechnologist. And Dr. Pomponi's research focuses on marine sponges, in particular, the development of sustainable methods for the production of sponge-derived chemicals with human health applications. And with that, I'm going to turn it over to Dr. Pomponi. Welcome.
Thanks, Dr. Scarpinato. Let me just see here about sharing my screen. It worked a little while ago. And there we go. Okay, so thanks very much, Dr. Scarpinato. Thanks to each of you for joining today. I just had a quick look at the participants list, and I'm really delighted to see some old friends there, people I've worked with in the past. So hope you enjoy the seminar today. When I was asked to talk about marine natural products drug discovery, I really wanted to focus on some of the work that our group is doing on sustainable supply options. And so I figured out a way to kind of work that into not only the seminar today, but also the topic or the title of the presentation. So I'm going to give you, there's a spoiler alert. So I'm going to give you the answer to that question. The answer is yes. So in this seminar today, what I'm going to be talking about is just a reintroduction to natural products, because I think probably many of you have either attended seminars by either myself or other members of our Marine Natural Products Drug Discovery Group at Harbor Branch, or you have a familiarity with marine natural products or natural products. I'm going to talk a little bit about marine natural product drug discovery, and then sponges, of course, because I always talk about sponges. Talking about a little bit about options for sustainable use of marine natural products for human health applications. And one option, in vitro cell culture production, which is what our research group is focusing on. And then some other challenges beyond and besides sustainable use. So I asked a colleague of mine, Peter McCarthy, who just gave a really great seminar yesterday as part of the Ocean Science Lecture Series, if he would share a slide with me, and he did. So I'm sure many of you are taking some of these medications that are derived from natural products, or you know someone who does, or you know, all taken aspirin. We've all taken aspirin from the willow tree. It's derived from the willow tree. Morphine is derived from the poppy. Taxol, which some of you may be familiar with. It's an anti-cancer drug. It's an anti-cancer drug, and it's derived from the bark of the Pacific tree. Ivermectin, a couple years ago, is actually an anti-parasitic. It was developed for veterinary use. And then, of course, we all know about penicillin. Was derived from a yeast, penicillin. And as it turns out, that natural products are quite common in the drugs that are administered today. In fact, in the last 40 years, more than half of all the drugs, 64% of all the drugs that were approved, had their origins in natural products, whether they're from terrestrial plants or microbes or even from the ocean. 58% of antibacterials, 78% of anti-cancers. The oceans are a really rich source of biodiversity. And that biological diversity provides the basis for a lot of chemical diversity. And that chemical diversity is really what gives us the potential for discovery of novel chemicals and bioproducts. So keep that in mind as I'm talking to you about marine natural products and sponges in particular, that basically that biological and chemical diversity really drive the discovery and the successes that have been made over the last, I'd say probably 50 or 60 years in the discovery and development of novel chemicals that have been or are in the pipeline to be developed as drugs. And in fact, the estimated annual value of marine-derived pharmaceuticals is now, over $2 billion, $2 billion. And there have been a number of marine policy briefs that have been prepared over the years that talk about the value and why they're so valuable. I'm going to highlight a couple of marine-derived drugs and tell you a story about each one of them. So the first one is Prialt, which is an anti-pain medication that's derived from a toxin that's produced by the cone snail. And the second is Yondelis, which is an anti-cancer drug that's derived from shallow water sea squirt or Ascidian. So as it turns out, because we may ask ourselves, okay, why is it that these marine organisms actually produce compounds that can be used as drugs? I mean, obviously, a cone snail isn't going to get cancer. Sponge doesn't get cancer. Why do they produce these compounds that have anti-cancer, anti-pain applications? So it turns out cone snails, so the source of this toxin that's been developed into a drug, are very sophisticated practitioners of what some of you may be familiar with combination drug therapy. So you may, for example, if you have cancer, you may be given an anti-inflammatory or in addition to an anti-cancer, or something that affects your immune system, for example. So cone snails actually have a number of chemicals in their toxin, multiple types of chemicals in their toxin. They're called conopeptides, and they act together synergistically to catch their prey. And the prey could be other snails, they could be fish. They can be lethal to humans if you're stung by a cone snail. And so we call this a toxin cobble. So the first is this thing called a lightning strike cobble. So the cone snails have this barb that shoots out and it'll strike the fish. So it'll immobilize it. It just strikes it and stuns it, paralyzes it. Then there's a motor cobble that'll inhibit the signals that are going, between nerves and muscles. So it'll again paralyze it. It can't move. The prey can't move. And then there's what's called the nirvana cobble. So that jams all the sensory circuits and it causes the fish basically to not really care that it's getting ready to be eaten. And in fact, I don't know if you can see my arrow or not, but at the left-hand side of the picture on the lower right, you can see the tail end of that fish that's being sort of swallowed. It's a siphon there in the cone snail. It's being swallowed, and it basically just doesn't care. So, but why do these chemicals have pharmaceutical potential? Well, as it turns out, some of these molecular pathways have been conserved throughout evolutionary history. And they're metabolic pathways and they're molecules that are also involved in human diseases. So cell division, cell death. So that's why something that causes cell division or cell death might be useful as an anti-cancer agent. In this particular case with the cone snail toxin, there are the toxins that are in that venom really interrupt a lot of channels like calcium and sodium regulation, potassium channels and things like that. So, oops, why is that not going forward? Let's see, let's try it again. So one of those compounds, one of those conopeptides has actually been developed into a drug, which is called Prialt, which I introduced you to a few minutes ago. It's a synthetic analog, so they don't have to collect a lot of snails and pull out that toxin. Chemists just have figured out a way to synthesize a chemical that's very similar to that conopeptide, and it's used as a drug. It's very potent, more potent than morphine, and it's used basically for end-of-life management of chronic pain that's associated with diseases like AIDS and cancer. Okay, let's talk about sponges. Sponges are the most prolific source of marine-derived chemicals that have pharmaceutical application. And in fact, the very first clinically available marine-derived drug came from a sponge, which you see in the picture on the far right. It's a sponge that was actually collected in the 1950s just off the coast of the Florida Keys. And from that sponge, there were some compounds that were isolated and then chemically synthesized and developed into drugs. And you're probably familiar, maybe, with at least one of them, Zovirax. I don't know if you ever get fever blisters, but if you get fever blisters, Zovirax treats herpes. So it's an ointment that's really useful and very, very helpful to treat herpes infections. And also another compound that came from that sponge is an anti-cancer agent called cytarabine. And then more recently, another sponge-derived drug, an anti-cancer drug, Halaven, was derived and is synthetically produced. It was derived from a sponge that was collected in Japan. Okay, why sponges? Okay, remember what I said, biological diversity is the source of that chemical diversity, which is then the driver for discovery of novel chemical compounds. Sponges are key components of many benthic marine ecosystems. They live attached to the bottom. They've developed sophisticated chemical signaling methods because they can't get away from predators. They can't get away from things that are trying to grow over top of them. They have to communicate with another individual of the same species when it's time for reproduction. And so they've really evolved a very diverse suite of chemicals that can be developed, can be tested for their usefulness as pharmaceuticals. There are more than 9,000 described species of sponges. They occur worldwide, from shallow water down to the deep sea. They even occur in fresh water. And it's been estimated that there's probably 25,000 species yet to be described. And these sponges are not only useful for the bioactive compounds, the chemicals that they produce, but if you had the opportunity to see Peter McCarthy's talk yesterday, they also are hosts of a diverse community of microbes, bacteria, fungi, archaea, and these could also be the source of the chemicals that are biologically active, and so very diverse. And you can see to the left of the picture of the sponge, it's an electron micrograph of bacteria, microbes that are inside sponges. Sometimes it can make up more than half of the biomass of a sponge. Sponges also produce, in the lower left-hand side, you see a scanning electron micrograph of what are called spicules. They're microscopic skeletal elements made out of silica. And as it turns out, these are actually sources of bioinspiration for structures, structures that provide, in fact, architects are using them to design structures that are very resilient to stress forces like high winds or earthquakes, things like that. But it turns out the biosilica that's found in sponges also will attract, in humans, if it's ground up into little particles and tested with human cells, it'll actually attract the cells that produce bone. So it may be useful in bone regeneration, bone remodeling, and so on. So these are scaffolds now for human tissue engineering as well. So a lot of products from sponges in addition to the chemicals. And it's been estimated that more than 75% of that global sponge biodiversity occurs in deeper water, so below 200 meters. Remember, I said we've got 9,000 described species, but because we've explored so little of the deep water, we really don't know how many new species there are, and the opportunities to discover novel chemistry, novel compounds from these deep water organisms. And in fact, these sponge grounds are widely distributed across the world's oceans. And this is just a picture of the Northern Atlantic Ocean. And every place you see those white dots are places where massive aggregations of sponges, either the same species of sponge or different species of sponge, occur. And this map actually was put together by my colleagues' project I was involved in that was funded by the European Union called Project Sponges. And part of the charge of that project was to discover some of these new sponge grounds and figure out what ecosystem services they provide, because sponges filter large amounts of water. They are also, as it turns out, excellent in terms of carbon, nitrogen, and silica cycling in the ocean. So very important in terms of not only drugs from the sea, but also in other ecosystem services. So the fact that we've got such great sponge biodiversity, and that below 200 meters it's even greater, really formed the basis for the development of the Harbor Branch marine natural products drug discovery program back in 1984. And we were very fortunate because we had at our disposal a fleet of research vessels and the Johnson Sea Link manned submersibles that allowed us kind of a leg up on our competitors in marine natural products drug discovery. So we could take the subs down to 3,000 feet deep and really selectively collect samples. We could see what it was that we were collecting. We could see how those organisms were interacting with other organisms in the environment, and very carefully collect those organisms. And this flow chart on the bottom was actually prepared by my colleague and the person who heads up our marine natural products drug discovery program at Harbor Branch, Dr. Amy Wright. So basically, we would collect the samples, and on the ship, we'd actually prepare extracts. So think of an extract as a cup of coffee. And so you're or I always like to say a sponge daiquiri, a sponge mojito. So you're grinding that up, sponge up in alcohol, and then filtering it out. And that extract contains lots of different chemicals. And what we're looking for, like in caffeine, the reason you drink, in coffee, the reason you drink coffee is for the caffeine. So the buzz that caffeine gives you. So we're looking for the buzz that's going to, say, kill a cancer cell. And so through a series of what we call bioassay guided isolation and purification, we give that extract to the biologists, in this case, Dr. Esther Guzman, who heads up the biology program, the cancer cell biology program at Harbor Branch. She figures out, is it active or not? She sends it back to the chemists. They start separating it out into the individual components. And then at the end of the day, hopefully, or sometimes it takes weeks, you get down to a single molecule that has that activity that you're looking for. And it might be a novel chemical with novel biological activity. So that's in a nutshell, the process. Okay, so what happens if we find a promising drug candidate? And let's just say it occurs at 2,000 feet deep in the Bahamas. And now we don't even have the ships or the subs anymore. So how do we get more of that to make sure that we have a supply that can go into preclinical development, clinical development, and then be available to treat patients, without going back and decimating the species, harming the environment? So there are several options for sustainable use. One of them, believe it or not, is controlled harvesting. So you could conceivably go and collect enough of the material. And for those of you ladies, and maybe some gentlemen, who use the Estee Lauder Resilience line of cosmetics, what you may not know, but if you look at the ingredients, I think it says marine extract or something like that. And in it is an anti-inflammatory material. So sometimes if you use cosmetics, your skin might get red, you might have a reaction to it. And so in the Estee Lauder Resilience line of cosmetics, they put in an anti-inflammatory material that comes from soft coral. And they actually have gotten permission to harvest at certain times of the year, just take little snips of the branches of the soft coral, which then will grow back. And they use that to get their marine extract for the cosmetics. Chemical synthesis, if the molecule that's produced is simple enough, not too difficult to chemically synthesize, then that's another option. If it's a microbe that produces the compound, and that microbe can be isolated and it'll grow in culture and you can scale it up. And when you scale it up, it still produces the compound of interest. Then that's another option, fermentation. I'm going to get back to the sea squirt and Yondalis, but aquaculture is another option. If the organism that produces the compound can be grown in aquaculture, then that's again a sustainable option. And the one that we're really focused on is in vitro or cell culture production of the compound. So which do you think, if you were a pharmaceutical company, you would be favoring? Okay, I see several hands raised. Not really. Okay. Chemical synthesis, of course, is the preferred option, because then the pharmaceutical company can totally control the process. They're not reliant on a hurricane wiping out the gorgonian, or the fact that the microbe doesn't grow, or you can't grow the cells in culture, or you have a disease in your aquaculture system and it wipes out the supply. So the preferred option really is chemical synthesis. In our group, I've been on a quest to develop a marine invertebrate sponge cell line for decades, really. And in this process, especially over the last 20 years, I've been joined by my colleagues at Wageningen University in the Netherlands. And this is just a introduction to the group. It's a group in the Wageningen University is headed by my colleague Renee Weifels. And we've had, and this is just a few members of our group, but there have been a lot of people involved in developing our sponge cell culture program over the last, say, 30 years at Harbor Branch for sure. Okay, so why do we want to develop a cell line? Well, as I mentioned, I think that this would be an opportunity to develop an in vitro production system for these sponge-derived products. And it also might give us some new models for bioproduct discovery, because it may be when we culture these cells, they may not produce the compound that we're interested in, but they may produce something else that's novel that we hadn't discovered before. I really am interested in why these sponges produce the chemicals that they produce. And also, now that we've got these models developed, we'd like to test some hypotheses that are related to evolution, developmental biology, ecology, systematics as well. And I'll talk a little bit about this in a couple minutes. We've actually been able to cryopreserve or freeze living cells that we can keep for future use. So we can preserve these cells from these species for future research. I'm happy to report that after decades of research, I really didn't think it would happen before I retired, but it did, and now I can't retire. We finally developed sponge lines, and we published this in 2019. So, but this path to this success had a lot of twists and turns and obstacles. There were a lot of challenges. Growing a marine invertebrate cell is not like growing a mammalian cell. All of the cell culture media, all of the methods that have been developed for mammalian cells that grow at 37 degrees C and at a much lower salinity. So we're constantly having to adapt our methods. But we've had a number of incremental improvements over the years, and then really one serendipitous event that happened. So for years, our model organisms that we've studied in our lab have been the sponge called Axonella corrugata, which is a reef sponge that occurs in Florida reef tract, and Disidia etheria, a sponge that grows pretty commonly on mangrove roots. But as it turns out, these sponges never, ever, ever respond. I mean, Disidia wouldn't grow, Axonella every now and then would divide, but it would divide on what I call a geologic time scale. I mean, if you wanted to create cell cultures and do in vitro production, you need to have cells that are dividing really rapidly. And, it might take 2 weeks for Axonella corrugata cells to divide. I'm not a patient person. 2 weeks is too long. So my lab manager, who's also my PhD student, Megan Conkling, said, hey, Shirley, we have this biobank of cryopreserved cells, more than 50 sponge species. We actually have thousands of vials of cells in the biobank now. Why don't I just test these nutrient media on some of the other sponges? And I said, sure, go ahead. We have nothing to lose. The model sponges that we've been working with have not been behaving. So she did, and lo and behold, one of the cells from one of the sponges, the sponge called Geodia neptuni, divided. I mean, they divided crazy fast. Now, this is a sponge I would have never selected as a model because it has a really thick cortex. It's really a bear to try and dissociate the cells, and the cells are teeny tiny little cells, not nice sized cells like you see in like mammalian cells, for example, or even the Axonella corrugata or the Dissidia cells, which in comparison are like gigantic. I mean, they're really tiny, but the Geodia cells are really tiny. So I mean, of course we were jumping for joy, we were all excited. So now we started, it's like, okay, well, now what? So let me go to the, okay. Oops, what happened? There we go. All right, I promise this is the only data slide. I just wanted to show you, illustrate that these cells really divide quickly. So on the top row are, let me go back a step. So on the x-axis, so the horizontal axis, you see hours, and on the y-axis, the vertical axis, are cell numbers. And so just to give you perspective, they start at about 1 million cells at the bottom and go up to, what's that, 10 billion cells at the top. And what we found was that with the sponge genus is called Geodia. No matter what Geodia we tested 3 different species, multiple individuals, they all divided. So very little variation, again, which is unusual for sponges because sponges have a lot of individual variation. And there's no doubt that for each species that we use, we're going to have to develop some novel nutrient media, to feed them. But as it turns out, Geodia turned out to be a great model organism, the genus. And so we tested, we've been testing 3 species of that genus. So on the far left of the graph, you see a column Geodia bereti, and there's a picture of that sponge that occurs in deep water in Norwegian fjords. So it grows at lower temperatures. And then the middle and the right-hand column are 2 species of Geodia that occur basically in the Florida Keys. Geodia species, that kind of lump that looks like it's got sediment all over it, occurs in grass flats. And the Geodia neptuni, that beautiful kind of vase sponge, grows out on the Florida reef tract. And so what you can see here, and just disregard the 3 blue lines, they're just individuals of the same species. At 22 degrees, so that top row, the Geodia bereti, which grows at lower temperature, which lives at lower temperatures, but these, with the exception of Geodia bereti, which also increased in cell number, they all increased in cell number and really in the first 4 hours, unheard of, crazy fast division rates. Geodia bereti did a little bit better at 4 degrees. So just put the cultures in the refrigerator, it did well. And even our tropical sponges did well. But in general, the tropical sponges reached a higher maximum cell density. So, again, pretty excited about this. But what if the sponge cells don't divide? So remember, I told you we were working with Axonella corrugata and Dissidia etheria for decades. We create a hybridoma. So the concept of a hybridoma is you take a rapidly dividing cell that doesn't produce a compound that you're interested in, and you fuse it with a cell that does produce a compound that you're interested in, but doesn't divide rapidly. So that's kind of the concept behind it. So on the bottom, in this little cartoon, you can see green ovals and red ovals, and the green ovals are Geodias, and the red ovals are the Axonella that never divided. You use a material called polyethylene glycol, and it causes the cells to fuse into a hybridoma. You culture them in nutrient medium, and ideally what will happen is that you'll get cells that divide and will continue to produce the compound of interest. So my master's student, Cassidy Dugan, is continuing to work on this, and she's actually been able to successfully get hybridomas formed, and we're getting ready to scale up the cultures so Dr. Wright can test them to see if they're producing the compound of interest. So we're pretty excited about that. What if the cells don't grow well in two dimensions? Remember, we're taking cells that were in a 3-dimensional organism, plating them out on cell culture vessels and expecting them to grow and do what they do in nature. We simulate their natural morphology by growing them in 3D. So my former graduate student and current research assistant, Elizabeth Urban-Gadamke, focused her thesis research on testing three-dimensional culture of sponge cells. And she used a number of different matrices. She used these things that look like little millipore filters that you see in the cartoon in the upper left. She used gelatin, so a type of gelatin, it's called agarose, and immobilize the cells in the agarose, and they would divide and then be immobilized and be able to grow in 3D. She also has them growing in little micro droplets, so little droplets, like think of like jello drops, but very tiny. And in fact, I'm pretty excited because we have these now scaled up and growing in what are called spinner flasks. So think about a kind of a beaker that's spinning, and you've got these little jello droplets with sponges in them, and the sponge cells with sponge cells in them, and the sponge cells are dividing. And then she also tested another method called creation of spheroids, which is a method that Dr. Guzman, our cancer cell biologist, has used a lot to be able to develop. She uses those for cancer cells to create cancer, like three-dimensional cancers she can use to test some of the anti-cancer compounds. With the idea being that they'll respond more similarly the way they do, in our body or in the ocean, than if they were just single cells. And Elizabeth was the first author on a paper that was recently published reporting the success. So we're pretty excited about this as well. So our ongoing work is to continue to establish some more sponge cell lines, continue to optimize our nutrient media, because I have an intern from Wageningen University who'll be leaving next week, and he has been testing some different nutrient media, and he had, again, just like everything that we've done with our sponge cell culture program, you got a lot of, just very frustrating because a lot of failures. It doesn't work. This isn't happening. And then just a few weeks ago, he tried an insect cell culture medium, and every species of sponge that he worked with, the cells divided. So we're pretty excited about following up with that work. So we're going to be scaling up our 2D cultures, our 3D cultures, and our hybridoma cultures to be able to document that we've got a system and proof of concept that we can produce these compounds in culture and then be able to scale up that production. We also have funding from the Harbor Branch Oceanographic Institute Foundation, especially licensed programs, and from the Environmental Protection Agency, which I just remembered I didn't put on my acknowledgement slide, for habitat restoration. So we're taking these cells, and instead of trying to produce a compound from them, we're trying to produce little sponges. So they'll aggregate and form a functional sponge. So that's another application of the work that we're putting a lot of effort into right now. All right, so I'm going to finish up by just talking about a few other problems here and challenges. I talked about adequate supply for development. I think we've got a number of options available to us, but there are a number of other challenges. Access and benefit sharing. As you might imagine, a lot of these organisms come from tropical environments, and a lot of these tropical environments are within the sovereign territory of developing countries. And in order to be able to go in, and I mean, you can't just go in and collect material that are the genetic resources of that country and remove them. You have to have permission, you have to be able to get access. And when you're to get access to that, sometimes you have to say, what are the country's going to say, what do I get out of this? If you develop a drug, what do I get out of this? Can you help us to do capacity building, raise up our scientific infrastructure, our educational infrastructure, so that we can actually participate in the discovery process? So access and benefit sharing is a big, big issue. Intellectual property and patent protection. If you don't protect your discovery, a pharmaceutical company is not going to develop it. If everybody has access to it, nobody is going to develop it. So that, again, is another really important factor to consider. And it's a challenge, it's a bottleneck. And it really depends on having very aggressive licensing and technology transfer programs within your university or organization, because otherwise you make really important scientific discoveries, but they're not going to go anywhere. And then finally, the timeline from collection to clinic can be just disproportionately long. For example, Yondalis. Oh, and I didn't explain that either. I wanted to talk a little bit about that Yondalis, because I was gonna mention that about this, how that was developed. That took more than 30 years from the discovery that it had some active compound in it to actual clinical application. And with Yondalis, what happened was PharmaMar, which is the company that developed it, later on in concert with Johnson & Johnson, actually went out and did massive collections. They collected from Cuba, from Florida, I mean, tons of these mangrove tunicates. They tried to aquaculture because, as it turns out, the sea squirt does very well in aquaculture. But in the end, what they did was they used what's called a semi-synthesis procedure. So they fermented a microbe that produces a chemical that's a precursor to actinicidin. And then it made it easier, a shorter process to chemically synthesize the molecule. So that was one where several different processes were tried. Wild harvest, aquaculture, fermentation, synthesis to get to that final product. So I want to acknowledge, and this is by no means an exhaustive list of people who've been involved in this project over the years. It really does take a village, and it takes a lot of dollars and euros to do the work. And so I especially want to acknowledge the members of my research group, Megan Conkling, without whom we probably would have never realized that we should try something different than those two model species that we've been working with. Elizabeth Urban-Gadamke, who's doing the work and has done some really nice work on 3D cell culture, and Cassidy Dubin, who's working on the hybridomas, but also my colleagues in biomedical marine research, John Reed, Amy Wright, Peter McCarthy, Chris Diaz, my partner in crime in sponge systematics, Esther Guzman and Tara Pitts, who've helped so much with microscopy and the other methods that we're using to evaluate, analyze our cells. Don Liberatore and the crews of our research vessels and the Johnson Sea Link submersibles, and then my colleagues from Wageningen University, seen here, listed here, and then our funding sources, NOAA, I mentioned the Harbor Branch Oceanographic Institute Foundation. Buy the license plates, that money that comes back it comes back to Harbor Branch Foundation, and that's what provides support for a lot of the research that we do. So if you don't have a shark tag or an aquaculture tag, please go out and buy one. The Gertrude Skelly Charitable Foundation, and then we had some funding from the European Union that supported graduate students Stephanie Monroe and Kylie Hess. I want to thank you also for your for coming today, for listening, and I'd be happy to take questions.
Thank you so much, Shirley. Just as a reminder, if you have a question for today's speaker, please use the Q&A button in the bottom of your screen. You can type your question there. And I just wanted to follow up on something that Shirley was just mentioning, that the drug development, no matter what you do, takes decades often, and is also very expensive, right? So in addition to the license plate program, if you want to donate to any of this, let us know. It is not trivial to develop a drug for pharmaceutical use and that's suitable for humans to take, since we have to minimize and want to minimize side effects, of course. So Shirley, you mentioned that some of these compounds that you and Amy and Esther are researching could have anti-cancer efficacy. And is there a limit, or do you see a limit on what type of diseases these marine-derived compounds could address, and could are there certain types of diseases? You mentioned, these compounds have a certain role in the animals that you're actually getting them from. So do you see any certain groups of diseases that could be treated via these compounds?
So that's a very good question, Karen. And Esther Guzman has over the years developed some really specific biological assays or tests so that we know not that it's just killing cells or killing a certain type of cancer cell, but how it does that, how it kills that cell, and it's called the mechanism of action. And when you patent and license a drug, pharmaceutical company is going to want something that's different from the way what's currently out on the market, or else they're not going to be able to market it. So over the years, Esther Guzman has developed biological assays that are focused on different types of cancer. So not just, like broadly against it. So she's looking at specific types of cancers. We can really pinpoint the type of activity that chemical has, but also anti-inflammatory activity, anti-malarial activity, antiviral activity. We have a very interesting compound that is an anti-inflammatory and also antiviral. And so, think about some diseases that are out there today that cause inflammation or an immune, attack your immune system. And if you not get a drug that can do both, that's very promising. So we're lucky because we have a number of collaborators with whom we work. And so those compounds that Amy and her colleagues have isolated over the years, thousands of them, are made available to our colleagues to test in different assay systems. So it's not just the work that's going on at Harbor Branch, but also in the labs of our colleagues worldwide.
It sounds like a very big, almost untapped arsenal of potential new drugs that are out there that all need to get tested, if they have activity and against what they have activity, which is exciting, but also is daunting in case of, wanting to test them. Okay, I'm not sure I'm going to pronounce this correctly, but do you plan to try culturing hexactinellate sponges, known as glass sponges?
No, it's a glass sponge. It's really interesting that you asked that question, because I've just been thinking about that over the week. So hexactinellids, these glass sponges, typically live in very deep water, below 200 meters, although in some parts of the world, they're like in fjords, and you can get them shallower than that, where it's really cold water that has upwelled from the bottom. Hexactinellids are kind of interesting because they're mostly made out of spicules with very little organic material. And their cells are a little bit different they're not kind of individual cells. They form what's called a syncytium, so kind of fused. But I like challenges. So we have a cruise coming up at the beginning of May. Is that what it is, Amy? Yeah, beginning of May. So I'm hoping that we can get some fresh hexactinellids then that I can try. So because Amy Wright has discovered some really interesting compounds from hexactinellid sponges that occur in a number of areas in the tropical Western Atlantic. And this one was found in the Gulf of Mexico. But I'm really curious to see if we can get those cells to grow in culture.
Very interesting. The sky's the limit, Shirley.
Yeah.
And I should mention, when Shirley mentions a cruise, this does not mean that she's going to go on a entertainment ship and go gambling. This is a very different type of cruise.
Yeah, there are no umbrella drinks on these cruises.
Yeah.
We're actually going on an expedition to the northwestern Gulf of Mexico off of the Flower Gardens Bank National Marine Sanctuary, and it's funded by NOAA. And it's a grant that Amy Wright got, and there are a number of us who are collaborating with her. And it'll be the first time actually that we actually looked at some sponges, and not only sponges, I'm prejudiced, of course, but organisms that occur not only within the Flower Gardens Bank, but outside outside of the Flower Gardens Bank National Marine Sanctuary, and on a little bit deeper water as well. And it's going to give us an opportunity also to look at sponges, for me, that occur in both shallow and deeper water and see what the differences are. And if in fact, they grow differently. We'll be collecting sponges and cryopreserving the cells from those sponges when we're out on that cruise. That's just one small task or project within that larger project on the research expedition.
I think everybody here can appreciate, hopefully can appreciate more now how difficult it really is to get to the point where you do have a pharmaceutics that actually then you buy in pill form or whatever form, right? And you start with something completely different, like a natural organism. Either you showed the willow tree for the aspirin, in your case it's sponges, right? But that's not, then you actually have to figure out, well, there are only so many of these organisms, you cannot just all kill them off to get your drug, as you mentioned. So what Shirley is doing is absolutely important in order to make enough of those drugs available in an efficacious way without killing all our sponges that we have in our reefs and our waters. So very important work that Shirley is doing. So we have another.
Actually, I just want to point out something else too. We have, because we were fortunate enough to be able to go for many years and do these expeditions, and again with the informed consent of the host country, we have samples in our freezer that we're able to go back to and use. So we've frozen this material. We've got extracts of those. We've made the little sponge daiquiris and Gorgonian daiquiris from all of them as well. But we've got the original material in the freezer. So it's a real treasure trove of material. And then, as Peter McCarthy talked about yesterday, he's got more than 19,000 microbial isolates that he isolated primarily from sponges that again can be tested. So it's a real treasure trove of for our current research program and future research as well.
Absolutely. Okay, Shirley, we have a question from a scuba diver and underwater photographer who is asking if you could speak some more about tunicates or sea squirts and if they have any benefits from medicine to medicine.
Right. So the one, in fact, probably one of the first, like after that sponge that from which we got the Zovirax, one of the first ones was that became really interesting and was this sea squirt called Echinacea turbinata. And you don't even have to be able to scuba dive to be able to see that. You can go snorkeling just off, in fact, mangrove roots in the Indian River Lagoon and see these little sea squirts there. And they're the source of that anti-cancer compound called yondelis. And so we have actually focused on, in our collection efforts, not only on sponges, but also on octocorals, soft corals, and ascidians or tunicates, because they are historically very rich in natural products with potential pharmaceutical applications.
Very interesting. One follow-up question from this underwater photographer who's asking if your research could benefit from a partnership with underwater photographers like the South Florida Underwater Photographers Society.
Well, if you have photographs to share with us, that would be great. It's really hard. I know from my perspective, I'm sent photographs of sponges all the time. And because sponges you really need a little piece of that sponge to identify it. And so it's really hard to be able to identify it like the organism just from the photo. However, where it could be really helpful is if, you see something unusual that you've never seen before. It might be something we've never seen before either. And it might be something where we could take a small sample, go back to that same location, take a small sample of it. I mean, assuming it's not in Tahiti or someplace like that, and take a small sample of it and test it. So yeah, that's a possibility.
Shirley, how did you get into the interest with sponges and marine organisms, and then particularly into looking for health-related treatments that come from these marine organisms?
I'm going to try and shorten this story. I wanted to be a nurse when I was growing up. My mother was a nurse, and I didn't want to go back then. You didn't have to go to college to be a nurse, but I was a good student. She said you have to go to college. When you finish college, if you want to be a nurse, you can be a nurse. So I went to college. got into kind of like a program where, was on a path to kind of do biomedical stuff. And then halfway through, I had the opportunity to go to the Virgin Islands for a summer program, a marine ecology program, and then I was hooked. And I went to graduate school, and then it's kind of an embarrassing story about how I got hooked in sponges. I took a class on systematics, so how things are named and organized, and we had to do what are called synonymies, kind of like the family tree of individual sponges, individual organisms. And sponges, you only had to do 6. And like if you did crabs, it was like 20. I'm like, all right, and I'm not a slacker. I mean, I'm never like that, but it was my second semester and I was like overwhelmed. I was like, all right, I'll do sponges. So little did I realize that the reason they only had to do 6 was because they were about 5 times harder than everything else to figure out their family tree. But I got hooked on sponges then. And so I really focused on sponge systematics and I really enjoyed cell biology right ever since I was in college. So I was really focused on using cellular, trying to come up with cellular characteristics to discriminate one sponge from another. And it just went on from there. I got my master's degree in that. And then I did my Ph.D. on cellular mechanisms by which some sponges can bioerode calcium carbonate substrates. And then I was in State College, Pennsylvania, freezing to death in December of 1984. And I got a phone call because I could identify sponges with someone from Harbor Branch saying, hey, we need somebody to help us identify some sponges. And I went through the whole thing. How many do you have? How are they preserved? How soon do you need them? He said, no. We're going to the Bahamas and we'd like you to come and help us identify some sponges that we're collecting for this program and drug discovery. And I'm thinking, geez, that sounds great. And actually it sounds wonderful. I had just shoveled snow. And I said, when is it? And they said, oh, we're really sorry, but it's next week. I said, send me a ticket. And that was it. I was hooked. That was December of 1984 and I'm still here.
That's a great story, Shirley. Okay, one last question before we're out of time. What is your recommendation if somebody wants to get into this area? If you have a student and who wants to go into this area, what should they be looking for? And also, if somebody from the community wanted to help with these kind of efforts, do you have any suggestions for that?
Yeah. So I mean, you don't have to have a PhD to do this research. There are positions available no matter what level of education you have. If you want to run your own program, you pretty much need a PhD to do that. But I'd say, the advice that my mother gave me actually wasn't an advice, it was a directive: go to college and get a degree. It can be an associate's degree, it can be a bachelor's degree, it can be a master's, a PhD, but get some advanced education. Biological sciences, chemistry, molecular biology, chemical engineering, any of those, are good. Get a good education, and then you can decide. You might have an opportunity to do an internship or to volunteer, and same thing like members of the community. Now that things have loosened up a little bit because of COVID restrictions are not as severe as they used to be. I believe we're at a point now where we're I think close to, if not already, taking some applications for volunteers as well. So there's an opportunity to volunteer. If you're a student, take advantage of internship opportunities. Now, our application period for summer internships at Harbor Branch just closed on March 1st, but we do it every year, and you have to be either an undergraduate or a graduate student. And if you're in St. Lucie County and you are interested in doing more marine-related work, and you're not in the Marine and Oceanographic Academy in the St. Lucie County school system, it's associated with Westwood, you might be able to transfer over into that it's public school. It's actually on the Harbor Branch campus, and the curriculum is really marine based. So those are some opportunities.
Great. Thank you so much, Shirley. So if anybody has any interest in participating, either volunteer or go to school and follow the instructions that Shirley just gave you. With that, thank you very much, Shirley. Very interesting topic on regulations on your cell line.
Thank you.
And I hope to see everybody back at our next Research in Action. Have a great rest of your day. Bye-bye.